we got to talk about the cell here. So chapter 3 is, of course, the next chapter. We're going to be splitting it up a little bit. So this particular lecture that you have, all these PowerPoints, the first part in gold is from chapter 3. We're going to talk about the plasma membrane of the cell. We're going to talk about the cytoplasm and organelles and the nucleus of the cell. Then we're going to actually go into chapter 6, and there's another little outline for chapter 6 once we get there. Then there's a chapter 3 part 2 lecture, which I'll be posting in chapter 3 part 3, which will cover gene expression, protein synthesis, DNA synthesis, and cell division. So those things we're going to be picking up with, but it should be easy to see when you move into chapter 6. And remember, the figure labels always have like a 6- something if you're in chapter 6 or a 3- something if you're in chapter 3. So it should be easier to be able to see. All right, so plasma membrane of the cell. Let's kind of recap what we've covered so far. We've discussed the atomic level. We talked about the molecular level in chapter 2. We talked about the tissue level at the end of chapter 1, and I told you we were going to spend some time now on the cellular level before we get to the organ level, which will be coming up in chapter 7 with the nervous system, which is our first organ system. So, first of all, the cell. We said that atoms give rise to molecules, molecules give rise to cells, and cells are the simplest level of organization considered to be alive. It is highly organized, and it has 3 major components: the plasma membrane, which we're going to look at, the cytoplasm, and the organelles, which literally organelle means little organ. So the cells themselves can live on their own, provided they have the proper medium. So they can live outside of the body. That's how we have tissue culture. Tissue culture, you take the cell, you put it into a special broth medium that's nutritive, and it has all of the nutrients the cell needs. The cell can live on its own and be a living thing by itself. So those little organs help it to do that. But let's start talking about the plasma membrane. So first of all, you've probably heard the term fluid mosaic used to describe the plasma membrane before. You've never heard this before? Never? Well, today is your lucky day, because we're going to talk about fluid mosaic. Now, what does this mean? Okay, we have who knows what a mosaic is? If you were going to tell me what a mosaic is, what?
Is it art? It is art.
Okay, so it can be art. You can have it in your kitchen, your bathroom.
It's
wherever.
like an image made up of many tiny little images.
It is, right, it's an image or some type of a composition that is made up of many different types of components, right? So sometimes people make a mosaic out of tiles, and they'll make like a cow or a face or something like that that's really nice. And the fact that the plasma membrane is a mosaic means that it has sort of a canvas, which is the phospholipid bilayer. And within this canvas, we have other components. We have proteins, sugars, cholesterol, and those types of things. The term fluid refers to the fact that the plasma membrane is always sort of moving. The phospholipids move around themselves. Sometimes these channels or these cell markers, they move around as well. And after we talk about the cell, I have a nice video I'm going to show you that will sort of illustrate these ideas that I'm describing for you, but we have to get through this part first. So the plasma membrane surrounds and gives the cell form. It's selectively permeable, and it is formed by a double layer of phospholipids, as we described earlier in the class. We have the polar head groups. You can see the round heads facing the outside and the round heads facing the watery inside environment of the cell. We have the lipid tails facing each other, just sort of like an Oreo cookie or a sandwich, right? So like Oreo cookie, the phospholipid heads, the phosphate groups would be the cookie, the lipid tails would be the soft, gushy center. So why is the double layer of phospholipids important? Why is it important to have a membrane? Just like any fence, right? It prevents, it basically prevents things from getting in and out that we don't want to. And it helps to, your pen just, oh, gee, that pen is really, your pen just completely fell off the table. I don't know, I just saw it rolling at me and thought, it was going, it was like running away from you. That was what was so funny. I just looked, I'm like, how did it start getting all that momentum to go? Anyway, so plasma membrane contains proteins which help to customize the membrane. I mentioned earlier that the red blood cells, of course, have a plasma membrane, and embedded in the plasma membrane of the red blood cells, we have what are known as glycoproteins that contain sugars. And interestingly, red blood cells are sticky. They're sticky. If you've ever cut yourself and you get blood in between your fingers, you know it does become sticky before it clots. And the reason for that is because of the cell markers that you have on the surface of your blood cells, in part, and also because of the clotting proteins. But these proteins do have markers that make each cell individual to you, and we call that your complement. So if a person, for example, needed an organ transplant, like a kidney or a liver, God forbid, the organ is really one of the most complex in terms of cell markers of all the organs that we have. It's definitely one of the most complex, and why it's so difficult to find an exact match. It's very hard. People wait for livers for a long time. But it's the glycoprotein markers that are embedded in the plasma membrane that give it this appearance. So anyway, also they provide structural support for the membrane. They serve as transporters. So some proteins, they go the entire way through the plasma membrane, and things can pass through the protein back and forth. When we talk about cell transport, we'll be talking about a lot of carrier proteins or channels. They can also be enzymes, which again help to speed up certain reactions. We'll talk about some of them when we get into metabolism. And again, they can be receptors to attract certain things. Like, for example, we have receptors for glucose, we have receptors for neurotransmitters, you know, like in the skeletal muscle cells. In the plasma membrane of skeletal muscle cells, they have receptors for a neurotransmitter called acetylcholine, which triggers a skeletal muscle cell to contract. And then again, identity markers, as we mentioned before. So carbohydrates can be in the form of glycoproteins, as we mentioned before, and glycolipids can also be part of the outer surface. Glycolipids generally impart a negative charge, which helps cells to sort of repel each other. Normal cells do not grow on top of each other. If they do, that means there's some pathological reason for it, and usually that is going to result in cancer. And then, as we said before, glycoproteins can serve as cell markers, which are also known as antigens. And in lab on Tuesday, we're going to be talking about antigens when we talk about blood typing. Some other things you can find associated with the plasma membrane are things like cilia. And in lab, we did see the cilia and how they appear under the light microscope. These are little hair-like structures that are embedded in the plasma membrane, but they're their own structure. They're not like microvilli, which are actually part of the plasma membrane that's wavy. The cilia, they are important for movement of substances over the surface of a structure. So cilia, they can beat together in unison to provide movement. So you'll find that in the respiratory tract and the reproductive tract. And the cilia contain microtubules in a 9+2 arrangement. What we mean by that is we have 9 pairs of microtubules, 1, 2, 3, 4, 5, 6, 7, 8, 9, around a central tube. And microtubules, again, are proteins that we're going to find not just here, but we're going to also find it in the skeleton of the cell as well. Microtubules you're going to see again and again, but they are a very important protein, and they are found making up the structure of cilia. Now again, I mentioned microvilli and how they're different, and we talked about it in lab as well. I drew for you that microvilli are actual waves within the plasma membrane itself. The microvilli have a different function than cilia, which are not the plasma membrane. The cilia are, in fact, their own structures, as we said. But the microvilli's job, having this waviness to the plasma membrane, is to increase the surface area for things like absorption to take place, or reabsorption if we're talking about the kidneys. So again, we saw this in lab when we looked at what? What did we look at? What tissue did we look at that we saw microvilli? Who can remember? I should say, who can remember? Isn't it funny how you can say the same sentence two different ways and people, you're like, who can remember? And then, who can remember? You know? Like, completely. No, so, what is the second way I said it, the more accurate? All right, so think about it. We talked about epithelial tissues, and we looked at cells that had cilia. When we looked at the trachea, right, the pseudostratified ciliated columnar epithelium. And also we saw the microvilli when we looked at the digestive tract in the simple columnar epithelium. That we did. So that's where we can find these things and where we actually did see them. Okay, so some cool things that the plasma membrane can do is they can take part in bulk transport. It can take part in bulk transport. There are two types of general umbrella bulk transport terms. We have endocytosis, which is where we bring things into the cell, and we have exocytosis. This is where we can take things and move them out of the cell, large things. So in terms of endocytosis, there are some more specific terms to describe the type of endocytosis. We have phagocytosis, which phagy means to eat, and it's phagocytosis we take in food particles or just large things that are going to be digested. For example, this here is showing us a macrophage, which is a type of white blood cell. Macrophages are large, that's what macro means, phage, eaters. These are going to engulf and go after bacterial cells. So you can see here that this macrophage forms what's known as a pseudopod, which is a false foot, to kind of surround this bacterial cell that you see here. And these feet will finally encompass the entire bacterial cell and then bring it into the cytoplasm of the macrophage, where it will be subsequently digested. So we'll digest it and destroy it, and that's what macrophages do. We'll talk about them today in lab. Okay, so bulk transport also includes another type in terms of endocytosis. We have pinocytosis, which is like cellular drinking. This is where cells take in non-specific, just water with solutes like ions, sodium, chloride, potassium, so forth, or sugars. And again, it happens the same way that phagocytosis did. And the last type of endocytosis is the most really specific type of endocytosis. This is called receptor mediated. Now, I like to take the word mediated and change it to controlled. So essentially what we have on the surface of the plasma membrane, we have some receptors to receive certain molecules. So for example, cholesterol molecules will get into a cell this way. The receptors are going to bind to cholesterol. Once the receptors are all full, that's going to trigger, that's going to control the invagination or the dipping inward of the plasma membrane to then produce this vesicle. So it's receptor mediated because it's once the receptors are actually full that the endocytotic process begins. So again, things like cholesterol can be transported into the cell this way. Some viruses can get into the cell this way. Also, by mimicking the structure of things that receptors are supposed to be binding to, like cholesterol. So that's endocytosis. We have phagocytosis, which is cellular eating, pinocytosis, which is cellular drinking. We have receptor-mediated endocytosis, which is where we take specific receptors and we bind to a particular substance to bring that into the cell. And once the receptors are full, that begins the endocytosis process. Okay, any questions so far about that? Endocytosis, bulk transport. All right, so exocytosis is the opposite. This is where things are released from the cell. So what kind of cells would use exocytosis? Well, for example, if we're looking again at the pancreas, okay, let's take a look at the pancreas. The pancreas contains those islet cells that produce insulin and glucagon that we talked about before. Now those are actually proteins, and those are made within the cell. So when we make the insulin, we make the glucagon, that has to be packaged in a vesicle which can be transported to the plasma membrane and released into the bloodstream. Otherwise, how else are you going to get it out there? So exocytosis will release things that are secretory, things that are going to be secreted from the cell. Secretory proteins like insulin or glucagon. All right, so any question about the plasma membrane, any of the topics that we discussed there? Okay, so we're going to talk a little bit now about the cytoplasm and its organelles. And I am going to go through this a little bit quickly because I know in anatomy you cover these things, but if you have any questions, please feel free to stop me. I don't want to breeze past and confuse anybody. So first of all, the cytoplasm, of course, is the fluid-like material, the jelly-like material within the cell. And why is it jelly-like and not just water? Because it does contain a lot of water, but it also contains proteins, sugars, ions, all of these types of things that make it more viscous or thick. So it's going to be more like apple jelly if you want to get a visual idea. And it also contains these organelles, which we're going to be reviewing. Within the cytoplasm, we have a skeleton known as the cell's skeleton or the cytoskeleton of the cell. The cytoskeleton gives the cell shape and structure and also provides a bit of help to move things around inside the cell. We have three components. We have actin, which is there for structure and support. We have intermediate filaments. I should have said before, the actin is also known as the microfilaments. We have intermediate filaments, which are intermediate in size, and microtubules. So the microfilaments and the intermediate filaments do provide a lot of structural support. Sometimes they anchor organelles in place. But the microtubules are really cool within the cell. The microtubules act a lot like train tracks. Remember those vesicles I just mentioned when we were talking about exocytosis? Those vesicles can't get from point A to point B without they don't just kind of float through the cytoplasm. They have to be guided to the plasma membrane where exocytosis can occur. And how does that happen? On microtubules. When I show you the video, when we talk about the cell, when I show you the video, you're going to see how cool it is because you'll see that there are actually these proteins that hold on to vesicles and walk along the microtubules to get the vesicle to where it needs to go. And Sometimes they fall off the microtubule and the vesicle is lost, believe it or not. Once I saw a man give a seminar on vesicles and vesicular transport, and I asked that question, and he said that they sometimes do get lost inside the cell, which is interesting. But when I show you the video, you'll get a really cool, you get a good visual and to see how that happens. So microtubules can assemble themselves quickly, they can disassemble themselves quickly depending upon where a vesicle needs to go. So it's very dynamic. And that's one thing you have to remember. These are very flat diagrams that you're looking at, but what's happening inside the cell is very dynamic. And that's why I do want to show you this video. We can get to it. All right, so next we're going to talk about organelles, starting with the nucleus. Well, we know the nucleus isn't always found in the center of the cell. Sometimes, like with adipocytes, there's so much fat in there that you have to push the nucleus out to the side, right? Or in skeletal muscle cells, we have more than one nucleus, and they are found out on the sides of the cell because we have so much actin and myosin in the sarcomeres. But the nucleus is enclosed by a double membrane known as the nuclear envelope. So we have sort of an inner part and an outer part which folds on itself. And we have little proteins that sort of rivet the two together. And through these proteins we have pores, which allow small molecules to enter and leave the nucleus. Interestingly, the nuclear membrane, the outer membrane, is going to be continuous with the other organelle we're going to talk about, the rough endoplasmic reticulum. And again, we have pore complexes that fuse the inner and outer membranes together so that small molecules can diffuse through. Large things like proteins or RNA molecules, messenger RNA molecules, they are going to have to be actively transported to where they need to be. So the nucleus is a very important organelle, contains DNA, also contains RNA. It also contains this little dark area right here, which is called the nucleolus. And the nucleolus is important for producing our ribosomal RNA. Ribosomal RNA is produced there in the nucleolus. Ribosomes are little protein factories, and they consist of two protein subunits. And you can see here's one subunit here that's a lighter purple and then a darker purple here. Now, interestingly, ribosomes they're not always assembled. They don't assemble until they need to make a protein. So in the video I show you, you're going to see how a ribosome comes together when it's time to make a protein. You'll also see in the video that I'm going to show you how the protein is made and how it folds and how quickly it actually folds. Okay, so ribosomes are where proteins are synthesized. They're found free in the cytoplasm, and they're also found in the membrane of the rough ER. Does anybody know why? Why they're found in the cytoplasm free, and why they're found also in the rough ER plasma membrane? Anyone know? Okay, so on a ribosome that is found freely floating in the cytoplasm, we can make a protein, and we can use that protein inside the cell, because we're not going to make a vesicle around that protein. But if we make a protein on a ribosome in the rough ER has membrane, right? So our protein that's made in the rough ER will be pinched off in a vesicle derived from part of the plasma membrane from the rough ER. And what's the purpose of this? Why do we put it in a vesicle? Because it's going to have to be transported on the microtubule to get to the plasma membrane, so through exocytosis we can secrete this protein. So to say simply, ribosomes free in the cytoplasm make proteins to be used inside the cell. Ribosomes free in the cytoplasm make proteins to be used inside the cell. Ribosomes that are part of the rough ER, make proteins to be secreted from the cell. Okay, any questions? All right, so the endoplasmic reticulum. So endoplasmic, endo means from within the cytoplasm. Reticular or reticulum, reticulated, these are all forms of the same word, kind of means spider web, meshwork, network-like. And we have two types of endoplasmic reticulum. We just mentioned we have a rough endoplasmic reticulum, which is rough because we have proteins that are embedded into the plasma membrane. And, we have ribosomes that are embedded in the plasma membrane. And the ribosomes here are going to make proteins that are going to be used to be secreted from the cell. So the ribosomes here are going to make proteins that go into the ER, and that protein will be pinched off in a vesicle derived from the membrane from the ER. Carried on a microtubule to the plasma membrane through exocytosis, we can release the protein. The other endoplasmic reticulum is smooth. It has no ribosomes in its plasma membrane. And depending upon the cell, the smooth ER will have different functions. For example, in skeletal muscle, we're going to have the smooth ER containing a lot of calcium. That's where calcium is going to be stored. In other places, the smooth ER is going to be used for steroid hormone synthesis. So it depends completely on where we are. Okay, so Golgi, the Golgi complex. Everybody always describes its structure as looking like a stack of flattened pancakes, one on top of each other. And the Golgi complex is kind of going to be the organelle that finalizes the packaging of the protein. So if we're making a protein that's to be secreted, we produce it on our rough ER, pinch off in a vesicle, goes to the Golgi. The Golgi kind of puts the bow on the protein, adds like a sugar carbohydrate chain or a lipid or something like that. And then we finally release from the Golgi the final protein, so it goes to the plasma membrane. So this is a great diagram right here that when you're studying and you want to remember the functions of all the different organelles, you can use this diagram to describe what's happening here. I really do like this diagram. I would put a star beside it. And I think that, if you approach your studying in the way where you're taking, all these narrative slides and you're describing it in the picture, it will make your life a lot easier and more efficient and effective studying. I think that works a lot better. So anyway, you have your, let's pretend we're gonna make insulin because we talked about that before. We have our pancreas, we have our cell, we've got the nucleus that contains the DNA to make our insulin protein. So that, we're gonna go to the rough ER. We're not gonna get into too many details at this point. We're gonna go to the rough ER ribosome. We're gonna make the protein there. Then we're gonna have a little vesicle pinched off from the rough ER, travel on the microtubule to the Golgi, where that insulin is gonna be finalized. Then a little vesicle is pinched off from the Golgi on the microtubule, travels to the plasma membrane, and then through exocytosis we can secrete the insulin. Okay, so that's one function of the Golgi. Now another function of the Golgi is to produce things that are also organelles called lysosomes. Now lysosomes are, we're going to see I think on the next slide, they're little vesicular organelles. They're little vesicles, little round circles that contain digestive enzymes. So they're kind of like your belly, you know, with food. They digest things. So how do we make it? Same way. We basically just, instead of making a vesicle that is going to travel to the plasma membrane for secretion for exocytosis, like with insulin, what we're going to do, we're going to make proteins in the we're going to get, you know, have our DNA in the nucleus. We're going to make proteins which are actually enzymes, which will destroy things. We're going to make them on the rough ER, package them in a vesicle derived from the Golgi, and instead of going to the plasma membrane, now that vesicle just can float around in the cytoplasm as a lysosome. It's not going to be secreted. But you've got the vesicle, you have the proteins, which are the enzymes that can break things down. So that's the two things that the Golgi does. And lysosomes, I do think, are on the next slide here. They are, as we said, they're vesicle-like organelles that contain digestive enzymes, and whenever they take in something like an old ribosome, for example, they do wear out, and the lysosome, what it'll do is it will release these what we call hydrolytic enzymes. Remember, hydrolysis is a reaction that breaks things down. When we use the term hydrolytic, that means that we're taking water and we're breaking things down. That's the kind of enzyme that the lysosome has. It'll break down all of these you know, proteins or bacterial cells or whatever, depending on what the function is. And also, lysosomes are involved in cell death, programmed cell death. Anybody know what the word is for programmed cell death? Starts with an A. Continues with a pop.
Apoptosis.
Yes, apoptosis. So actually, when a cell needs to die, okay, when it's old and worn out, or if, for example, in development, okay, we're born with more neurons than what we need. And as we learn fine motor skills, the other neurons die off. Likewise, in fetal development, you know, initially we have webbing between our fingers, and those cells will die in order to give us our digits, so we have enough movement. So what happens is the cells, when they're programmed to die, they will open up their lysosomes and all of these enzymes and basically digest themselves from the inside out. And they commit suicide this way. So it's kind of both interesting and gross at the same time. Okay. And lastly, we have second to lastly, we have the peroxisome. Peroxisomes are also organelles that are vesicular, and these guys contain enzymes which are oxidative. So they're going to eliminate free radicals, and you're going to find a lot of peroxisomes in places like the liver. And actually, one of the ideas of aging, one of the theories of aging is that our cells start to have less and less peroxisomes. And because of that, we have difficulty getting rid of things that aren't so good, free radicals and stuff in our bodies. And that's why you get these like skin spots and stuff. Also, it's why people tend to not recover as quickly. Like I can't say you'll relate to this, but you know, like if people drink a lot, and you know, maybe when you're younger you can bounce back from a hangover a lot faster, but as you get older you can't, and that's part of the reason, because the peroxisomes aren't necessarily as plentiful. All right, lastly, in terms of organelles, and then I'll ask for questions. The mitochondria, the mighty mitochondria. Everybody knows that this is where you produce your energy. Right? Also known as ATP. We have ATP in our bodies. That's our energy currency. That's what we can use. Our cells use ATP. Now, the mitochondria, you may remember from anatomy, is a very special organelle. Why is that? Oh, we drink a lot of water. Why is mitochondria so special? Doesn't have its own nucleus. It's got a what? Not on the wall, but I think along what you guys are saying, I know it doesn't have its own nucleus, but it does have its own DNA. Right, it has its own DNA, which is interesting because you think the nucleus is where the DNA is, so I get where you're coming from. But it's different for the mitochondria, and I'll explain it. And then, Nastasia, you were saying it has its own wall, and I think what you're thinking is it has two membranes.
Yeah.
Right? Good. So those are two interesting things. Okay, so there is that idea that's out there that the mitochondria is a pretty autonomous organelle, and the reason why they think that perhaps, this idea why it's autonomous is because they think early in human development, back before we had multicellular organisms, we had a bacterial cell that was engulfed by another cell that was a single-celled organism. And that bacterial cell was able to remain alive within that initial cell that engulfed it. And the bacterial cell gave the cell that engulfed it amazing properties to be able to produce energy very efficiently and effectively. And the original cell gave that bacterial cell the safe environment in which to live. So they called it a symbiosis, whereby each organism benefited. Now, what are some evidences for this? Who can remember? What are some evidences that the mitochondria could possibly be two cells that sort of combine together? We already mentioned it has a double membrane, and the inner membrane is wavy, which actually resembles very closely what bacterial cell membranes would look like. The second is it has its own DNA. But the DNA isn't like our DNA in our cells where it's, it's a helix, but it's not like a straight helix. What it is, it's circular. It's circular, which we call plasmid DNA. Plasmid DNA is a round DNA form that you find in bacterial cells. And the other evidence for this, anybody know? Anybody think of it? Because it has its own DNA, but what else can it do?
Reproduce?
Yep, it can reproduce. It can divide on its own by something called binary fission, which is specifically how bacterial cells divide. Bacterial cells basically will just split in two. And again, that process is called binary fission. Mitochondria do that as well. So mitochondria is a really special organelle. Interestingly, the DNA that's found in the plasmid DNA that it contains is mostly from your mother, and that's why they use maternal mitochondrial DNA. They try to use to trace to an individual's mother for various reasons, criminal cases, or sometimes just forensic analysis trying to figure out who was the last living relative of somebody, those types of things. Okay, so now what I want to do is show you the video that I had been mentioning to you so that you get an idea of how dynamic the cell is inside, and I also want to try to review some of the concepts that we talked about today and give you more visualization, and I think this video should do that for you. used to. How do I get a new window? I'm used to a Mac. I don't know why. I'm always telling them this. How do I get a new window on here? Nobody knows. You guys are like stone silent. No, you're not really. You're pretty
You
good.
need a new window.
Yeah. How do I
Go
do it?
down to the internet button and left or right. No, the internet button.
Here.
Yeah.
Left.
I believe so.
Open your town.
Wait, yeah.
Open
where
this
is it?
down.
There you go.
Yeah. Thank you. Sorry. It's just I'm not used to the PC. Let me see. Okay, so I'm gonna turn off the light here. I think the volume's up. Just to make sure. I don't want it to be too loud. The music's pretty. Okay. Let's see here. Oops. I don't know if I want to do the narrated. I don't think I want to do the narrated version. I'm just afraid it might be a little bit more overwhelming. Because then you'll think, well, what are those words? Did she say that? I didn't say. So those are red blood cells, and these are white blood cells up there. They were going through a blood vessel. Phospholipids. So notice how the heads like move around. That's what makes it so fluid. See how it's very fluid. It's always moving. This is part of the cytoskeletal framework. So, you know, the different proteins. And that's the protein filaments assembling, and you'll see them disassemble. So there's your intermediate and microfilaments and microtubules. That's an enzyme that'll clip off pieces. That's a microtubule assembling. Like I said, they act like train tracks. And disassembling depending on where it needs to go. That's the vesicle, and that's the protein that walks it along the track. So if there's going to be a protein in there that is carrying to the plasma membrane or to another organelle. You get a chance to see the proteins also. That's a ribosome, and watch, it's going to be making a protein. See how quickly it folded? Primary, secondary, tertiary. Look at that. Primary, secondary, tertiary. So quick. That's a ribosome coming together, and that's the mitochondria. You can see more proteins being made over there. Still walking. Golgi. See how the vesicles, they come in, and then the proteins are modified, the vesicle comes out. That's exocytosis of a protein. You can see how that works. Mix it. This is a white blood cell, and look how it stretches out, and it will actually make its way out into the tissue spaces. We're going to talk about that today. It's going to go out into the tissue space where it's going to like destroy all the bacteria. Really cool. So anyway,
Roughly look at this stuff under a microscope.
well, Unfortunately, I don't have any way to have a live specimen beyond the microscope, but if I did, I'd love it. We have that new electron microscope, and I'm still in the process of trying to learn how to use it because it's very complicated. But even there, I can't use a live specimen, but I could show you like some, like bone fractures and stuff like that. That's kind of what I want to show people under that. I think that would be really cool. But unfortunately, my knowledge, I'm not there yet. So I have to show this. But anyway, I hope that sort of recapped everything a little bit for you, what we talked about, some of the highlights I mentioned. So do you have any questions about anything? Other than can we go now? Yeah, you can go. If you don't have any questions, go ahead. I'll see you guys upstairs, some of you